Honors Biology, third quarter review, part 3, support for Darwin's ideas. Remember that Darwin said that living things change over time. Change over time as a result of variation, differences in the population, and competition for resources. We're talking about natural selection resulting from those that are best adapted, surviving and reproducing better. And as a result, things change to adapt to their environment. So keep in mind, when we're talking about support for Darwin's ideas, we're talking about how living things change over time and how we know that. The fossil record. Fossils are impressions and remains of organisms that lived in the past, and they're only found in sedimentary rock. Sedimentary rock is formed gently. The sediment covers the animal. The bacteria eats away the soft parts, leaving the hard parts behind, and the hard parts get replaced by minerals, leaving an impression of the animal or plant or any other organism. They're only found in sedimentary rocks because igneous and metamorphic are too destructive to preserve any remains. Keep in mind also that most things that die don't leave behind remains. Most things that die are completely lost as far as any of their body parts. It's only the rare organism that leaves behind a fossil. So as you can imagine, digging in dirt and rock, we don't have all the answers as to every transitional species that ever existed, the 2 million plus species. However, we do have a lot of information about what existed before present time. Now keep in mind, new layers cover older layers, and this is called relative dating. Relative dating means that the older layers are going to be found lower than the younger layers, which are found on top, and that's because of the way the sediment gets placed. All we know in relative dating is that lower is older and higher is younger. Fossils within layers show that a succession of organisms have populated the Earth throughout a long period of time. And there's a general trend in the fossil record, going from simple to complex, that we're going to talk about in a second here. Before that, however, remember that absolute dating involves finding out the actual age of a igneous rock using radioactive isotopes, things like uranium and thorium and other very long half-life elements that are radioactive are used. And using a ratio of radioactive isotope to decayed substance, you can figure out the age of the rock. For more details on that, review your notes on Chapter 22. Now, the fossil record has a general trend from simple to complex, and you should be aware of that. In the oldest and lowest layers of rock on this planet, dated at about 3.5 billion years old, we find bacteria, and that's the only form of life we find in those very ancient rock layers. Remember that the oldest rocks on the planet are 4.6 billion years old, so we think that Earth is around 4.6 billion years old. So once again, Earth 4.6 billion years old, bacteria 3.5 billion years old is the oldest known fossil of stromatolites. After that, the next major life that we find in the fossil record is single-celled eukaryotes. Still very simple, but we have eukaryotes. And remember that we think that the mitochondria and chloroplast were once free-living bacteria because they replicate independent of the rest of the cell. They have some of their own enzymes, they have their own DNA, so they might have been once free-living bacteria. The next thing that we find in the fossil record that's fairly interesting are the invertebrates, or animals without a backbone. Things like trilobites and other animals are very common during the Paleozoic. The next major type of life we find in the fossil record are fish, and the Paleozoic is often called the time of fish or age of fish. After that, the next thing we find are amphibians, and after that reptiles, and then in Mesozoic era we find the birds and mammals starting to evolve. Remember, there's 4 different time periods, and you have to know those 4 different time periods. It's Precambrian, most common form of life is bacteria. Then we have the Paleozoic, which is the age of fish, the Mesozoic, age of reptiles, and the Cenozoic, age of mammals. Here are some fossils. We have lots of fossils in Colorado, petrified wood, etc. Using the fossil record, we can try to trace back and piece together what happened in the past. And what we do is we make a family tree of this. Here we have a family tree of the elephants and some of the fossils that we find in the past, and these are dated millions of years ago. Keep in mind that the dinosaurs went extinct 65 million years ago, and that's a key date that you should know. And then after the dinosaurs disappeared, after they died out, and the leading hypothesis on this is that a large asteroid hit the Earth around 65 million years ago, kicked up a dust cloud, blocked the sun, collapse of the photosynthesizers that rely on the sun. The animals that required the most energy, the dinosaurs, larger animals, are the ones that were most effective, and affected and they died out. Now, what evidence do we have supporting this? Well, we have a giant crater in the Gulf of Mexico from 65 million years ago, and we also have a thin layer of iridium only found in asteroids at 65 million years ago as well as all the dinosaur fossils other than the ones that evolved into birds disappearing at around 65 million years ago. You have a tree of life diagram in your notes. You can review that. But the important thing to be able to do to read one of these is that any branching point is a common ancestor to whatever comes afterwards. So right here is the common ancestor to these guys here, the stegodons, which are no longer with us, and these guys here, which include some that are still alive, Asian elephants and African elephants. So this is not a stegodon. This is not an African elephant. It is a common ancestor but to both of those. So that's how you read this. This is a more distant common ancestor, and another branching point further back in time, a more distant common ancestor with more differences in DNA and more differences as far as bone structure. We have a directional or selective change in horses that increase the size of the horse, directional selection. A very long time ago, after the dinosaurs went extinct, the evolution of mammals to occupy the niches that were once occupied by dinosaurs starts to become more common. Over time, horses have increased in size. All right, this is a key fossil you should be aware of. It's Archaeopteryx. Archaeopteryx is a transitional fossil linking birds to reptiles in the past. It's basically a reptile. It has teeth like a reptile and scales. However, it also has feathers, and this is thought to be the missing link between birds and reptiles. Archaeopteryx, transitional fossil linking birds to reptiles. And we started watching a movie on this. This is Tiktaalik. This is the earliest known amphibian to have the four-limb template that we all have today. All right, moving on to biogeography. Different organisms developed in different areas, not based on who's best adapted to the environment, but what their ancestors gave them. For example, rabbits are better adapted to Australia than the native species. When rabbits came over, when humans brought them, they basically took over the place. There's thousands of them everywhere, possibly millions. However, because the Australian continent never evolved placental mammals, it broke off from Pangaea before placental mammals evolved. The only things that were trapped on there were simpler animals, the monotremes, including things like the platypus, as well as the marsupial mammals that have a pouch. And though marsupial evolution resulted in things like kangaroos, sugar gliders, things like that, no placental mammals are found in Australia. The explanation for this is that the ancestors didn't have a placenta and it never evolved on Australia, so that's why there's no Australian placentals. Now, even though they look similar, sugar gliders and flying squirrels look very similar, they are not the same, and there's a lot of differences internally, especially during for reproduction. Analogous structures. Analogous structures are structures that have the same function, similar functions, but they have different internal forms in development, and there's no close evolutionary relationship between those analogous structures. An example would be fish body parts and fins. A fish fin and a dolphin fin on the outside look somewhat similar. They have a large surface area for pushing through the water. However, if you look at the bones internally, there's not very much in common. And as a result of different DNA sequences that code for different types of structures internally, there's a lot of internal differences that implies a more distant ancestor. So if it's an analogous structure, no close evolutionary relationship, but might have the same function. Another example would be bird wings and butterfly wings. They have the same function, but they don't have the same internal structures and no evolutionary relationship that's close. Now, homologous structures are different. They have the same internal structure, they have the same embryological development, but they might have different functions in the adult. For example, the human arm, the cat front leg, the whale front flipper, and a bat wing all have very different functions. However, they all have the same internal structures. They all have the same kind of bone arrangement. And as a result of that, same DNA sequences making the same bone structures, and less time that would result in more differences between those organisms, they're fairly closely related from an evolutionary sense. So When we're looking at these structures, we try to determine the relationship between organisms, not just by looking at the bones, but also comparing DNA, fossil record, all those things kind of come together. Homologous structures, once again, same internal structure, might have different functions and evidence of a common ancestor. Vestigial organs are structures that serve little or no function. And it's also evidence of change over time. Why else would snakes and whales have remains of pelvis and leg bones if they didn't have walking ancestors? Now, it's not that they disappeared because of disuse. Keep that in mind. It's not Lamarckian. What we're talking about is mutations that didn't detract from the survival of the species. If you don't have working eyes, if you're living in a cave, well, there's no selective pressure for that because eyes are not a survival adaptation for a cave where there's no light. The human tailbone isn't one bone. It's 4 vertebrae fused together that match up with the bones of other animals with tails. Comparative embryology seeks to look for comparisons between different species during their development in the embryo stage and look for clues as to what might have happened in their past. For example, fish, reptiles, birds, and humans all have gill pouches when they develop. Now, of course, humans, birds, and reptiles don't have gills as an adult, but the idea behind Darwin's statement, descent with modification, is really brought home with comparative embryology. Now, while a gill pouch will turn to gills on a fish, we've modified these embryological structures from our past to become the inner ear and lower jaw of humans. And these different structures are modified in different species, though they all have the same starting point, which is a gill pouch. And same thing with things like tails. Even though we don't have a tail as an adult or even as a born child, we do still carry some DNA for making that at the early stages of our development. Molecular biology, this is a comparison of DNA. Remember that the more closely related you are, the more DNA you have in common. Just think you and your parents have the most common DNA versus everyone else on this planet. And you can take that idea and extend it beyond the species. Now, of course, the universal genetic code for all living things is DNA and RNA, and we can use that as a basis of comparison to find out how many differences. Keep in mind, the more distant the relationship with another organism, the more mutations that have accumulated over possibly millions, if not hundreds of millions of years, and that would result in a lot of different structures being created. Remember, DNA codes for those structures. And this is something that Darwin never knew. He never knew about molecular biology or DNA, and this is the best evidence for evolution and relatedness of species that we have. Cytochrome and hemoglobin are used, but for the test, just remember DNA can be compared to determine relatedness. Here we have hemoglobin being used. Humans with other humans, the same amount of hemoglobin amino acids, no difference, and of course humans are the same species. Rhesus monkeys, 8 differences in amino acids, so a little bit more accumulated mutations resulting in differences in the protein over time. Mice, chicken, frogs. Eventually you get to lampreys where there's so much difference that resulted from so many mutations over so much time that there's very little in common between the hemoglobin of a lamprey that carries oxygen and a human. That would imply a lot more distant relationship between humans and lampreys than humans and monkeys or humans and mice. Building family trees, we can use these DNA homologies as well as comparative anatomy and fossil record to put together these family trees. And we talked about this already. The branching point is the common ancestor to both those things. So when we look at a family tree and say, oh, we evolved from fish. Well, yeah, somewhere back in a very long time ago, we did have a common ancestor with fish. However, that was probably like 500 million years ago, 500 million years ago. We also have a common ancestor to primates. However, that's a pretty distant common ancestor. The most recent common ancestor are the chimps, 6 to 7 million years ago, and more distant for the other primates. Artificial selection. Artificial selection is humans changing a species over time. This is the mustard plant. We're growing those in class. And using different parts of the mustard plant, we made all these different vegetables. Brussels sprouts, kale, red cabbage, cauliflower, broccoli. All these are modified mustard plants, and that's all been done by artificial selection. That was a big influence on Darwin. He was kind of figuring if human beings over a fairly short amount of time can dramatically change a species, could nature do it over a longer period of time? Natural selection in action. Now here's some more examples. In addition to the peppered moth, you should be aware of insecticide and drug resistance. Insecticides don't kill all the individuals. Some of them have the genetic ability, not caused by the insecticide. It's natural to their populations to survive insecticide use. They have a gene for that. And the survivors pass on the traits. Keep in mind, this is not one bug turning into two bugs. We're talking about some survivors producing some offspring that eventually become a population. And then over time, if all the survivors have the genetic trait to survive the pesticide, you spray them and then they don't die. That's evolution. The genetics have changed over time, which is the definition of evolution. The result of natural selection, or us spraying those pests and unfortunately not killing all of them. In order for all the insects to die, you'd have to find a pesticide that no insect was genetically able to fight off. We've had the same problem with our drugs. Our antibiotics are less effective today because antibiotic resistance in bacteria has been selected for accidentally by doctors and the medical community. When you don't take the entire course of antibiotics, if there are some survivors with a genetic resistance, they survive, they reproduce, and now the antibiotic doesn't work. So there's a constant battle to try to discover new antibiotics to fight off the evolved bacteria that have fought off our earlier antibiotics. Penicillin is very much less effective nowadays than it was 50 years ago as a result of this natural selection. All right, so we're going to end with this for part 3. Remember that genes mutate. This is the source of all new traits. The other source of variation in a population is sex. Sex spreads variation. Mutated genes cause the new traits. So remember that. Individuals are selected. Now, individuals could be selected by nature or by artificial means, by humans, but some survive better than others and pass on their traits to more children. And then once the entire population has that trait, that's called evolution. Remember that natural selection happens first, and then when the population has the genetics that reflect the survival traits, that's evolution. This ends part 3 of your third quarter review.